Welcome to The Upside with Lou Bassanese. I am fortunate to have back with us the CEO of Gain Therapeutics, Gene Mack. Welcome back, Gene.
Thanks, Lou. Great to be back. Thanks for having me.
Listen, man, let's just hit it right on. Who would have thought you could release data that indicates exactly what we thought, that you have a paradigm shifting treatment potentially for Parkinson's, and the market just doesn't seem to get it. They're reacting the wrong way. I mean, what's your response to that? Besides, this is hard.
It's not atypical to announce good news and have the world sort of not understand it.
Yeah.
Particularly when it comes to biotech. And that situation is probably exacerbated by the fact that this is the first time this has ever been shown in Parkinson's. So I think a lot of investors maybe don't know what to do with this data, but it is enormously positive for us.
Yeah. And that's what I wanted to get on to talk with you about too here. Let's get into some of the data, some of the things that stand out to me. Let's start filling in some gaps here if we can. And then also just, provide an update on like what's next, because I think that's the key thing in biotech is always data, and the data keeps getting better here, not worse in any way, shape or form. So talk to me about the GCase substrate that you said it was a predetermined endpoint for the FDA recognized. Yep. So talk to us about that and explain that for the layman.
Yeah. So first thing is we thought it was important to mention in our announcement that this was a pre-specified endpoint in the phase 1A protocol, in this study's protocol. The importance of that is we were not cherry picking this data. This isn't us looking through every subject that was studied, their blood and their cerebrospinal fluid, to try and find something that's working. This was something that we were hoping would work and that we were hoping to see, and indeed we have. So it was important to number one, say we were going to do this as a pre-specified endpoint, this analysis, and with fingers crossed. And then the other important aspect of this is that it has not been seen in Parkinson's disease before. Some of it is because the technology is available to us now that maybe wasn't sensitive enough years ago when it would have been relevant to look. Some of it is that not a lot of drugs target this pathway, so what's the point?
Right.
But for us, this particular lipid substrate was the thing that the KOLs were guiding us towards. If you saw GCase activation, as we did last year around this time when we did our healthy volunteer study, and we were like, are we hitting the GCase target? Yeah. We got that, but there's nothing to measure after that because these are all healthy individuals.
Well, let's talk about that. Where we're at from last year to this year. I mean, those were healthy patients, like you said, so you can't monitor any anecdotal other biomarkers because they don't have the disease. So now you're getting that with patients that have the disease. So talk about the significance of that.
Now we can look at, okay, does modulating GCase even mean anything? Now, again, it's still early days in this study. So the patients clinically, we've seen some improvement. We talked about that in their UPDRS scores that we presented about a month or two ago. They continue on drug. And now we're looking at these other predictive indicators that those MDS UPDRS scores will continue to be durable and not slip on us and deteriorate, like a background Parkinson's population.
So correct me if I'm wrong. It seems significant to me that these patients were on drug only for 90 days, and you saw this, this activation happening. It's never been seen before. Is that accurate to say?
Well, the biomarkers, those should start to react. Between, if not now, it probably would have been a head scratcher for us. We probably would have started to try and take blood from patients that were in the extension study that were back on trial to see what we could see was going on, maybe beyond 3 months. But within 3 to 6 months, you should start seeing some of these things start to move. And we did. What you won't see is the clinical presentation of this stuff because the body doesn't, the biomarkers are predictive of the biology getting right.
Right. So it's the precursor to actually seeing the physical.
That's why they're relevant. That's why biomarkers help with drug development, because if you can align the biology with the disease progression and then interrupt that biology, then the FDA starts to say, okay, you're interrupting that biology. It's so well correlated to symptom presentation that you don't even need to show us that.
Yeah, that they know that that's what's next. So let me ask this as well.
You always like to reaffirm the what is already known, but it becomes a much more valid marker for efficacy, and that's what we're hopefully establishing here with glucosylsphingosine.
Yeah,
and That's the mouth. That's the letter salad that this toxic substrate is.
I'm not even going to try and pronounce it, but I wanted to ask this. To get this biomarker data, correct me if I'm wrong, you have to do a spinal tap, right? You're going...
Yeah, well, so blood evidence is great.
Yep.
But we've mentioned this before. Blood can be noisy, and it can tell you a lot about things going on in the peripheral body. And a lot of the things that we find in brain are found in different concentrations in the body. And when you try to, it's one layer away from the direct activity in the brain. So we only care about CSF right now. We only care about what's happening in the brain. That is the most proximal point and the most relevant biomarker.
So talk to me about what that means for the patient. What does it entail to get that biomarker data? You're doing
So a reduction in glucosylsphingosine, glucosylsphing for short, a reduction in glucosylsphing that we're seeing should help. So an accumulation of glucosylsphingosine is associated with mitochondrial dysfunction, lysosomal dysfunction. There are alpha-synuclein aggregation. So a lot of this downstream sequelae that contributes to the storm of symptoms in Parkinson's are, again, part of this.
They originate with that. But I mean, just the actual physical test, right? To instead of a blood draw, walk me through, you had, correct me if I'm wrong, you had 79 patients decide to volunteer to stay on drug. Yeah. Doesn't that mean that they have to voluntarily take an uncomfortable test to harvest the biomarker data at a periodic basis? Is that right or no?
Well, voluntarily and have to are kind of mutually exclusive. But we've asked them to. We don't know what kind of commitment we're going to get, because a lot of times they'll say, just to be in a real world scenario for a second, we know who has basically said, yeah, we'll do it. But on the day of, are they really going to be comfortable getting a needle in the spine? They can refuse at any time because it is voluntary in the extension phase of the study. I don't know that we'll learn much new. I think we have a lot of biomarker evidence with what we saw in healthy volunteers multiple times with GCase enzyme activation. And now to be able to see in patients, yes, the logical conclusion from that should be a decrease in one of these substrates, this particular substrate, glucosylsphingosine, then that is a key to understanding that GCase modulation is the right way to attack this pathway.
Right.
And we are getting to the pathway. We're not just activating GCase to no end.
Yeah.
We are seeing the biology play out along the lines of our hypothesis, hence the pre-specified endpoint in that phase 1B. We need to look at
So it's right, because the FDA approved the trial design and that is the pre-specified endpoint. But let's go, you just said it. You said you have, you're confident in the data that you've got. You've got additional data. Why, as in a layman, why wouldn't you share additional data here, or when will it, reasonably be shared?
So there's two important considerations there. There's the CDAs that we have with potential strategic partners. Where there are some, where we're under CDA, and there are certain things that we promise to share with partners.
So they get first look at it before you can put it out publicly.
Yeah, and quite frankly, we can only really talk to folks under CDA with some of this data, because if we present too much of it in a press release, that destroys our ability to present it at a medical conference. They take those things seriously. They don't appreciate when sponsors unload all the data, because then what's the point of the medical conference?
Right. It takes the exclusivity away and the appeal from the medical conference.
And investors, some sponsors try to get away with that and loophole it, and they hope to get under the radar. We're not doing that, because we need the medical society. We are doing cutting edge science at Gain. This is groundbreaking. There are no findings in the literature such as ours, because none have ever been reported. Doesn't mean there's not drugs out there that can't do what we've described.
Right, but this is, you said it, it's one of a kind, the first time that this has happened.
This is the first time this has happened.
Yeah.
So that allows us to be contextualized first. And the KOLs, before everybody goes in and rushes to the KOLs to ask them what they think about it, we should probably have a conversation with them to see what they think about it and work through with the field. Yeah. And we're trying to do that because what we are doing hasn't been done before, and we need to get it right.
Well, with that in mind, you simultaneously announced that you're going to have a KOL call on January 6th. Talk to me about the two doctors that are going to be participating in that KOL call and what their backgrounds are, and that which gives them the authority to really speak, as not the company officially, to the significance of the science of what you're seeing.
Dr. Roy Alkley and Dr. Peter Lansbury are among world experts in Gaucher's disease and a lot of this type of lipid. Now, Gaucher's is very close to GBA, and if you remember, our program started out as a GBA program. So we need to engage with both Gaucher's folks and also Parkinson's folks, because this particular lipid, glucosylsphingosine, is implicated in both very degrees. These two world experts, and when I say world experts, there's probably a handful of folks on the planet who understand the lipid biology as well as they do, that you can get in touch with.
Right.
And so these guys are going to help contextualize for the general public, investors, whoever's willing to, whoever wants to dial in, contextualize what this finding should mean for Parkinson's disease pathophysiology. And so we're going to try and contextualize all that on January 6th with experts who, we rely on for some clinical trial. Obviously no sponsor works on a drug development program without engaging several teams of KOLs to make sure you're getting the right feedback so that you can really enrich your clinical trial for success.
Right. So everyone should be paying attention. It's open to the public. You can register. I think the link was in the press release.
The information was on the press release, and we encourage everybody to dial in and learn something about a disease that impacts really all of us in some way, shape, or form. And we are making progress here.
Yeah. So moving beyond that, what are some other catalysts that everyone can expect? I mean, again, I always say there's a lot of noise in the market, always, every single day. We're going to look back a couple months from now, and this will just be a blip on the radar as you guys continue to progress. But what are some other catalysts that investors can be expecting as just signifiers of your continued progress? Yeah.
So the first thing we're going to do is go to the JP Morgan conference in January, so we can meet with all our potential partners and investors, and make sure everybody's on the same page with the type of data that we have and its implications. And then we've got to get straight to phase 2 planning, which is already underway. And we got to start making arrangements for that and announcing that we're going to open up clinical trial center. At some point, we'll discuss the progress that we've made with FDA on our IND filing, and also opening up centers for our phase 2 study. That's all happening early next year. And also the continued cadence of, what I think maybe fell flat was we're going to continue to report on, I think some of the questions we've gotten for our folks is that where's the UPDRS data and all that? Well, it wouldn't make any sense to reiterate what we just said. Those scores are still there. And now we want to see how durable they are. But that's going to take time.
Takes time for it.
We'll be reporting out, on regular basis, as the data comes in on UPDRS scores, how these patients are doing. So we'll be able to watch this trial all through the beginning of next year as we're planning phase 2. So I think there's a lot of catalyst-rich opportunities for us to continue to provide evidence in support of GT-02287 and really the exciting data that we've already got. And by that point, we will have shared most things with our partners. Whether or not something comes out of those discussions is,
that's in their hands, not necessarily yours. So you did also reveal on this in the press release that you have a stronger balance sheet capitalization than people may have anticipated. So you're fully funded through next year. Talk to me about just where's your confidence level now, right? Like we talked the first time when we did, what are the risks? Do you see any new risks that are coming after this data, or is your confidence even better than it was when we talked prior to this biomarker data?
I think we have a disease modifying drug for Parkinson's disease. And every day I get more certain of that. And impacting this particular lipid substrate just tells me we need to get very, very serious about preparing this company for a phase 2 study. So that's planning is underway. Organizational charts, things have to change at Gain to prepare, but we're getting ready for it. It's exciting.
Yeah. So it's more of an execution now. Stay on plan, head down, keep working at what you've been working at. And, more importantly, you're changing patient outcomes most likely based on what we're seeing here is the takeaway I get. Am I wrong in that assessment?
We like I said the last time we got together, we've seen these anecdotal improvements in these patients. We want them to be durable. They need to be durable. And hopefully this gives us evidence that those anecdotal improvements wind up being much more durable and also measurable separately from UPDRS. Yeah. Some other aspects of Parkinson's we haven't been able to get to with the drugs that are currently available, we might now be able to get to and measure and create better endpoints and help the guys coming behind us with drugs to complement what we're doing. Is one drug going to stop Parkinson's? yeah, I hope ours is it, right? But it would be hugely naive of me to think how many disease states are fixed by one drug except for insulin, and there's a bunch of things you need to support that.
Right.
So I don't anticipate Parkinson's being fixed by one drug. I believe ours will be the first disease modifying one. I believe we'll need help. I believe we'll need help to further slow and stop the symptoms. And we're going to be working behind with our backup compounds too, that are in some cases more potent and might be able to deliver more efficacy. This just creates more work for us, though. It's great.
Yeah, it's good. I mean, these are the positive things. It's a positive development. More good data, hopefully, to come. And look, would love to touch base with you again in the future on upcoming announcements as the market slowly digests this and hopefully figures out and connects the dots on how big of a deal this could be and how much upside there could be.
Yep, the data has never, since I've joined the company, the data has never been as rich and as robust, and the balance sheet has never been as strong in the 2 years I've been here, at least. So
that's a great position to be in to kick off 2026. So.
Well, there's a lot of wood to chop, but we're getting there.
Yeah. Well, Gene, as always, I thank you for your time and making it understandable to us laymen that don't necessarily have the science background as you and some of the other investors. So appreciate it.
All right. See you soon.